Hi everyone, this is Fredrik Wormling again. Thanks for watching this video. My research group at the Karolinska Institute in Stockholm, Sweden studies cancer and autoimmunity, and we're trying to identify novel drug targets and understand biology that's relevant for these diseases. And in this we use a lot of CRISPR and CRISPR screens. And this is the first video then in a set of videos where I'm going to show you how we approach analyzing CRISPR screen data. And in this particular video I'm going to show you how we process data files that you get from the MAGeCK software or script. And of course this is related to CRISPR screen data, so you sequenced your cells where you performed your screen, and then you're analyzing the NGS data with MAGeCK, and then the type of data set that you get out are as you see here on the screen. And I'm going to remove myself here and then we'll talk about it. So the type of data you get looks something like this. You have a first column that tells you about the gene ID, so very important. I'm going to label the ones that I find most important. Then you have the number of guide RNAs that were included to target this gene, and this was using Brunello in a human cell line. So this is 4 guides per gene. And then you're going to see we have a negative data set and a positive data set. So in this particular case, of course, negative and positive implies if the guide RNA was enriched or depleted when you compare these sort of samples. In this case, this is a traditional dropout screen. So the CRISPR library was introduced into the cells, and then we took an early and a late time point and just look at genes, how they were, or sort of how the genetic heterogeneity is changing over time, showing us if any of these genes are important to the cells or if the cells actually like if you get rid of them. So in the particular setup here, any negative scores, negative log fold change implies that the cells don't like if they get rid of this gene. So there's less of the cells with this knockout genotype or targeted genotype at the later time point compared to the early. And the positive one are the opposite, right? The cells like if you knock these genes out, essentially. And the columns besides the ID, gene ID, that I usually look at is the FDR value. So this is the statistics for both the negative and for the positive. And then we have the log fold change negative and log fold change positive. And for some reason they are the same number. So you typically just need to look at one of them. What I typically do is to look at the last one and then just call it log fold change, like this. And now I highlighted all of the columns that I find important here. So typically I save these files, but I would typically remove the ones that are not highlighted here for the type of analysis that I spend most of the time on, like this. And then I would filter them, so data filter, allowing us to look at sort of the negative with the most negative statistic value or FDR value, sorry, the lowest FDR value for the negative and for the positive. So we can sort this back and forth to look at the genes that are relevant here. What I typically do is then to use a cutoff of 0.05, but you can of course adjust that. I usually label them with the color, making it more easy when you sort of rapidly look at this data set. So these are the ones with a positive FDR value, sort of a significant FDR value for a positive effect. So the log fold change is positive. So if you knock these out, the cells in this case are increasing over time. And here you saw that this was roughly 17 genes, which is not uncommon that you see fairly few in this domain or in this category. While when you look at the negative FDR ones, so these are genes when you knock them out, the cells don't like it. They are usually many more. So and this relates to this sort of core essential type of genes that cells really need. So a lot of translation, transcription, and these sort of things, as you'll see. So I think usually there's more than 1,000, somewhere between 1,000 and 2,000 depending on the cell line. And I also usually label these just for ease of following it. So now we're more or less ready with processing the sample the way I usually would do it. I live in Sweden and my computer is sort of set for Sweden, so we usually use comma instead of dot for separating numbers. So I usually press Ctrl F, replace dot for comma to make my computer more happy. And I also usually don't think you need this many decimals. So I usually press format cells and then say maybe 3 here. to make this data set a little bit more easy to work with. But now, more or less, again, I keep the original file, but this is the one I would sort of go and use for further analysis. And the genes now that's been labeled with red are the genes that are significant in our readout, and those are the ones that I'm in the later videos in this playlist are going to show you how you can use different analysis tool to go into. So with that, I'm putting myself back here. I thank you for watching this video, and I hope you learned something, and I hope you'll watch the rest of the video. So thank you.